Hello, welcome back to the afternoon session of day 4 of BBE 2021. And a reminder that today's sessions are being hosted by the Responsible Interactions research theme. And just a reminder about how to comment and ask questions. If you're watching on YouTube, then please add any questions or comments to the comments section on YouTube, and they'll be fed through to me, and I can relay them to the speaker after the session. So it's a great pleasure to introduce our next speaker, Professor David Benjamin from Columbia University in the USA. David is the founding principal of The Living, a design and prototyping studio, an associate professor at Columbia Graduate School of Architecture, Planning and Preservation. David's work involves exploring new ideas through prototyping at the intersection of biology, computation, and design. And the reason for inviting him to this session in particular, because he could easily have spoken on one of the other days of this conference, the reason for inviting him to this particular session is his impressive portfolio of large-scale prototypes. And I was intrigued by this description of The Living from their website, quote, "The studio welcomes rapid change, embraces design with uncertainty, develops rules rather than forms, and designs with unknowable forces," which I think is a fascinating description. So I'll hand over to David to tell us about his work.
Great. Thank you so much, Ben. And it's incredibly exciting to be here at the summit. The work that all of the students and faculty at HPBE are doing is so forward-looking and inspiring. I love all the thinking and the physical prototypes and the compelling visual material. And I'm super honored to be presenting alongside the other visionary speakers at the event from HPBE and beyond. So I will maybe share my screen now.
Yeah, Ruth, maybe do you need to stop sharing and then David can share his? Great.
Great, so I'll share desktop and then present. And so hopefully that's coming through for you.
That looks great, thanks.
Great. So I thought that today I would like to talk about some of the work that we've done, as you just mentioned, Ben, at the intersection of computation, biology, and sustainability. And I'll describe some of the prototypes that we've been working on that involve some challenges in scaling up, some challenges in building things out in the real world, and some challenges in testing ideas out in public. So I think you'll see that this work involves biotechnology and the built environment, but it also involves other loops of ideas, materials, and society. In other words, we're integrating ideas about biotechnologies and microbiomes and things like that with ideas about things like construction and resources and climate change and just generally change over time. So first, I'd like to acknowledge the incredible team that I work with. All of our work in our studio is collaborative, and these people here on the screen, individually and collectively, literally bring it to life. And so I'll start by just describing that I believe buildings are living organisms. They breathe and pulse. They inhabit complex ecosystems of species, technologies, and culture. And understanding buildings requires understanding these vital signs and ecosystems. This kind of thinking allows us to expand the definition of environmental sustainability, and it allows us to expand our design ecosystems as architects. Along these lines, I'll describe our design ecosystem at The Living, which includes a variety of interconnected forces, and it involves a hybrid of the familiar and the new, the ancient and the cutting edge, the technical and the social, the tangible and the atmospheric, the practical and the critical. So the topic of our work often involves sustainability and climate change, and our approach often involves new possibilities at the intersection of biology and design. Of course, architects and other designers have been inspired by biology for hundreds of years. You may recognize this book here. But I believe, as many people have already demonstrated in this summit, that biology of today is different. It's now possible to grow a cell alone on a glass slide instead of inside an organism. It's now possible to visualize neurons firing inside of a live animal in real time. This is work of one of our collaborators. It's even possible to cut and paste DNA and bring to life creatures that never before existed, such as yeast that produces an anti-malaria medicine. And as of just a few years ago, as you probably already know, with the demonstration of CRISPR and things like gene drives, it's now possible to redesign or even eliminate an entire species very quickly, essentially molding evolution itself. In addition, it's now possible to apply the latest techniques of computation, such as computer vision and machine learning, to processes such as biological growth. Biological functions involve hundreds of dimensions, but if they can be simulated inside a computer model, then they become a more actionable part of the design process. Yet, the more you learn about biology, the more complex you see that biology is, and the more you see how much we still have to learn about it, at least in my opinion and experience. And in this sense, design with biology may involve things like design with a black box, design with forces beyond our complete control, and design with uncertainty, the kind of things that Ben just mentioned. This new version of biology calls for a new method of design. And in the past few years, we have developed 3 specific approaches to biodesign and 3 categories for projects that I'd like to describe now. So the first category is what I call biocomputing. And I define this as using actual living organisms to process information and solve problems such as, for example, reducing carbon emissions. So an example of this project involves our work with Airbus, the large airplane manufacturer. Airbus is interested in new ways of designing and making airplanes, kind of futuristic vision. But they are also interested in starting with tangible demonstrations. So the vision involves producing airplanes that are lighter weight and that therefore consume less fuel and have less carbon emissions. And the question is how to do this, how to make a new way of designing and making airplanes that are a lighter weight. And in our collaboration with Airbus, we started with this somewhat banal and forgettable component inside an airplane cabin called a partition. It's the dividing wall between the seating and the galley in a plane. And like all components in an airplane, it must undergo incredible challenges of certification to be integrated into today's flying planes. So this is one of the many tests that a component like a partition must pass before being allowed into the plane. And you can see a partition, the kind of partition that's in today's airplanes, undergoing what's called a 16G crash test. You can see how much force this partition must withstand just by looking at the kind of crash test dummies seated here. So we selected this component to design in a new way, in part because even though it may look kind of boring and forgettable, it's actually a challenging part to design in a lightweight way with traditional engineering approaches. The partition can only be 1 inch thick. It can only attach to the fuselage, the main structure of the airplane, at 2 points above and 2 points below. It must hold the fold down cabin attendant seat, which you saw in the crash video. And that's an asymmetrical load. The seat is essentially hanging off of the partition and pushing it in one direction. And also this smaller shape inset shown in the diagram I'm showing now it must be removable. It's called a stretcher flap. And if someone gets injured in the seating area of the plane, you have to be able to put them on a stretcher, bring them kind of around the corner and out the door of the plane, which means you need to be able to remove part of this partition, which adds an extra challenge to designing a good structure for this component. So it's at this point that we turn to an actual living organism. At this, in this case, it's called Physarum, also known as slime mold. So this is some interesting work and research by others. You may be familiar with some of this paper. of how slime mold grows in complex adaptive networks. These networks are efficient and robust. They're efficient in that they use a small amount of lines to connect the dots, which in this case are sources of food for the Physarum. And they're robust and redundant in that if you remove one of the lines, the system as a whole or the organism as a whole can route around the problem generally and still keep all the dots connected. So here we used a version of biocomputation to create an algorithm that models the logic of these complex adaptive networks, not necessarily the form, but the logic of these networks to kind of connect the dots of the partition, to connect the points where it connects to the fuselage, but to connect the points where it connects to the fold-down cabin attendant seat and other equipment that it must hold. And we used a version of artificial evolution in the computer to generate, evaluate, and evolve literally thousands of different design options, thousands of different ways of solving the problem of creating a new airplane partition that is both lightweight and strong. And here we can use some techniques of computation to kind of number crunch all of these different design options. So every dot on this graph is a different design where we want the lowest value on the x-axis. So we want minimum weight, and we also want the lowest value on the y-axis, minimum displacement under structural load. And we can get a set of designs that are mathematically good at meeting this challenge. And then in this project in particular, we, like many processes in nature, reran this kind of system or this logic at multiple scales. So the first step, which I just described, is taking the problem outline shown on the left and creating this overlapping kind of lattice of bars shown in the center that kind of connect the dots of the partition. But then in turn, we can take each bar or line segment shown in the center and in turn create that bar out of dozens or even hundreds of micro lattice bars shown on the right. And this allows us to create a partition not out of standard stock material, but out of a very highly calibrated set of micro lattice bars. In fact, there are 60,000 micro lattice bars in our version of the partition, essentially showing that this is a design approach that could not be achieved by a human alone or without this kind of biocomputation process. Like many of our projects, we aim to kind of explore some future technologies and some future ideas, but also to build it out in the real world. And in this case, we have printed in 3D printed metal the airplane partition. So this is some of the components that we created using a kind of new material, a proprietary alloy developed by Airbus called Scalmalloy, to create a partition that is basically the largest metal 3D printed airplane component to date. Here you see a version of the entire full-scale partition. And this is intended to fly in today's planes. So here is one of our latest digital simulations of the performance of this partition under load. And here's a corresponding physical test. And you can see some nervous Airbus engineers in the background watching this test. We're pulling at 24 times the force of gravity on the partition. And it's essentially supposed to bend but not break and then go back into shape. And you'll see here that in essence, it passed the test. There was one component with the crack, but this was a component, and that was just a headrest falling off, so that's not a problem. And the component with the crack was actually part of the traditional manufacturing, not this new biocomputation process. So this component is currently being certified to fly in today's A320 planes. It's about 45% lighter than the state-of-the-art partition that's flying today. It's about 8% stronger. And if this partition was kind of rolled out to all of the A320s flying today, this could eliminate 1 million tons of carbon per year. So the second approach I want to describe is what I call biosensing. And this involves using actual living organisms again, but in this case to detect and make visible the invisible conditions of the environment. This is a public interface to water quality in New York City and the East River. And it involves a floating network of lights, which are these transparent tubes which have dynamic LED lights above water and a variety of sensors below water. And our initial versions of this prototype involved electric circuits, digital sensors, the kind of material of the so-called Internet of Things. But our more recent prototypes have involved biosensors. And by this, I mean using actual living organisms, in this case, living mussels, the shellfish, to detect conditions of the environment. So here's a kind of sped up video of mussels in a tank in a lab, and you can see they're kind of pulsing around because mussels open and close their shells a small amount to eat food and to basically breathe dissolved oxygen. And it turns out that the rate and amount of opening of shell is an incredibly sensitive detector of pollution and other stresses in the water. And this allows us to take living mussels, glue to one side of a mussel's shell an inexpensive magnet, glue to the other side of the shell basically a $2 Hall effect sensor, a magnetic field sensor. And for just a few dollars, we have a better tell-all sensor of stresses in the environment than, say, a $10,000 dissolved oxygen sensor. So this is good for, a low-budget prototype, but more broadly, it allows us to think of possibilities for the built environment more generally. In other words, we're interested here in combining artificial intelligence with natural intelligence, combining the best that AI, the best that kind of computation has to offer, this holy grail of AI, with the best that biology and nature have evolved over thousands or millions of years to do incredibly sophisticated things that are in some sense like computation. And here is a version of this installation kind of out in the real world. And you can see the color of the lights changing because there's a kind of tipping point in the data, because the water changed from being a little bit worse right now than last week to being a little bit better right now than last week. So it's a way of visualizing the invisible forces that are important to our urban environments through biosensors. The third approach I want to describe is, I think, probably familiar to a lot of this audience, and we call it biofabricating. And by this, I mean using actual living organisms, in this case, as tiny factories to manufacture renewable or low carbon building materials. So this is a project that was part of an installation at the Museum of Modern Art and MoMA PS1 in New York City. And it was part of a competition. And our original idea for the competition was to see if we could take the Earth's natural carbon cycle, this endless loop of growth and decay and regrowth, and create a version of architecture that was a little different than our typical buildings. In other words, maybe we could take low value raw materials like waste materials, spend a small amount of energy converting those raw materials to building blocks, create a useful structure, and at the end of the useful life of that structure, take all of that matter, all of that physical stuff of the building and return it to this loop of the carbon cycle rather than putting it in a landfill where it might sit for hundreds or thousands of years. So this is, approach that's very related to circular economy. We were developing this project several years ago, kind of in the early days of the articulation of circular economy, and but it shares a lot of that vision of circular use of both technologies and physical materials. So how do we do this? How do we explore this diagram of a good idea for buildings? Again, we used a living organism, in this case, mycelium, which is, as you probably know, the branching root-like structure of mushrooms. And here, through some microscope videos, you can see how these networks form, and you can see nutrients pulsing through the kind of filaments of mycelium. It creates these incredible forms. But we were interested not so much in the form of mycelium, but in the function of mycelium, because, as you also probably know, you can combine mycelium with agricultural waste, not the high value part of agriculture like corn kernels, but the low value part of agriculture like corn stover, chopped up corn stalks. Combine these two things together, mycelium and agricultural waste, and in just about a week, it grows together into a solid object, as you can see here. And this is a really interesting approach for growing building materials as opposed to making building materials in other ways. It's been experimented with for a variety of things, including packaging materials. And our idea, this was several years ago, was to see if we could create a new kind of load-bearing architectural brick. This was in 2014, and since no one had made large-scale architecture out of this material before, we had to do a lot of material testing. Here we're testing a single brick in a strength of materials lab at Columbia University. We're testing small assemblies of bricks, getting a readout of their stress and strain over time with a certain amount of load. And this is where our kind of testing had the challenge and the friction of making something out in the real world, because our structural engineering partners at Arup were doing some calculations to make sure our structure would be safe under hurricane force loads of wind. And we quickly realized that there is no drop-down menu item for mushroom brick in structural engineering software. So we had to collect the data through physical tests, put it into a custom material profile, and then this would allow us to simulate the performance of a structure that was about 40 feet or 13 meters tall. In some of the first tests, we had these red areas, which meant that under these really high force of winds, we would have too much displacement. And so we had to, at this point, design at multiple scales at once. We were designing the material itself. What was the ratio of mycelium to agricultural waste in a single brick? How much did we chop up the corn stover in the brick? How long did we grow it in two separate phases of growing? So we're designing the material itself. We were designing the shape of the brick as the second scale, and we were designing the form of the 40-foot tall structure as the third scale. And after a few different iterations of designing and calibrating each of these scales, we got to a point where we had a model that we thought was acceptable and safe. Another interesting thing about this material is that it's different on the inside than the outside. So if you cut open a brick, as shown here, you can see a kind of skin on the outside, which is very naturally water resistant. If you pour water on it, it will just shed off without absorbing. But if the brick is cut, then it's much more porous on the inside, and it potentially will absorb water. So this was relevant to us because we had to make a structure out of bricks without cutting them on site to make them fit into our structure. So it affected our assembly method. And here we turn to a kind of custom computational model to solve two problems at once. We had a fitting problem, meaning every course of bricks was a different length. How do you fit bricks of fixed dimensions into these different length courses? And we had a stacking problem, meaning that every brick needs to sit with 2-inch overlap on top of the two bricks below it in order to hold together as a structure. And our computational model allowed us to create a semi-complex geometry and solve these two problems at once. How do you assemble 10,000 bricks in a way that we solve both the fitting problem and the stacking problem? So in the summer of 2014, we arrived on the site of MoMA PS1. This is the outdoor courtyard of the museum, and set out to assemble the building. And here, we combined in our kind of design ecosystem what I sometimes call the forces of creativity and labor. In other words, we were on site, we had to figure out how to do something that no one had done before, and we combined the expertise and thinking and problem solving of Columbia University graduate students who knew a lot about form and computation, and New York City brick masons who knew a lot about stacking objects to make structures. Neither one alone had made something like this before, so we brought into the design ecosystem this kind of collaboration and discussion and level of creativity and labor. And here I want to note that everyone who worked on this project was fairly paid. So in other words, I'm describing how a design ecosystem of testing things like biotechnologies out in the real world quickly comes into friction and intersection with a lot of other interesting things, including labor, materials, creativity, expertise, waterproofing, et cetera. So here is a view of the completed structure, and you can see it kind of in the context of the glass and steel buildings of Manhattan in the background and the traditional clay brick buildings of MoMA PS1. It kind of frames the natural environment. We were interested in the kind of atmospheric potential of this material, not just the technical potential. What would it be like to stand inside a structure made of this material? What would be the qualities of light and shadow and pattern? But the ultimate test of any project at MoMA PS1 in the summer as part of this program where many experimental structures have been built in this courtyard, the ultimate test is the ability of the project to host a good party. Because every Saturday of the summer, 5,000 people come to the courtyard to hear experimental electronic music. And so here was the first Saturday, both thrilling and terrifying. When people came into our structure, they were walking inside it, they were smelling it, scratching it, even climbing it. And when darkness fell, we breathed a sigh of relief that this was probably going to work. So although it made us a little bit nervous, this was entirely fitting for us to test our new idea, our material, our design thinking, to test it out in the world, in society, in culture, in a way out in public, rather than only on a lab bench or in the fenced off corner of a construction site. And here's some views of people interacting with the structure in the context of other functions and culture, like a music concert. Of course, as there often is these days, there was a kind of engagement with the public through a shorthand type of commentary, and that was interesting for us to monitor as well. Like, how would the general public perceive this new idea? Would it make sense to them? Would they appreciate it? What do they think about mycelium and biotechnologies and compostable architecture? Then at the end of the summer, we took apart the structure. It was, commissioned really just for 3 months in the summer. So we took it apart, took the bricks, crumbled them into smaller pieces, combined them with a kind of a new ecosystem, which included yet other organisms, worms and bacteria in a composting site. And in just about 60 days, all of the stuff of the building, all of that matter, returned to soil through composting. And then we took that soil and gave it to New York City for tree planting and also for community gardens, essentially showing that this is a building material that is non-toxic enough to eat. Again, we were looking at very specific materials and processes and a site, but we were thinking about broader ideas that might be applicable elsewhere. And one of the ideas we started thinking about here was that maybe as architects and designers, we can start thinking about designing to disappear as much as we traditionally think about designing to appear. And now I'd like to describe how, so if that was 3 different approaches we've been thinking about, I want to describe how these kinds of approaches and research directions are deliberately open-ended. In a way, the projects are never finished. The goal is not to just create a pavilion and photograph it and say, here's the idea, but it's designed to generate new questions and new possibilities. And with some of these kinds of projects and experiments, they can grow into new projects. So for example, with the project Hi-Fi that I just showed, our mycelium bricks were alive during their formation, but then they were dried out and they became dead, inert, and predictable during construction and occupation, just like most building materials. But as many of you have already been talking about in the summit and thinking about, we were just fascinated by the possibility that the bricks could be literally alive. What if we didn't dry out the bricks? What if they didn't become dead and inert? What if building materials were literally alive? What might our built environment look like? Would these materials and our walls and our structures be able to have some of the same properties of living organisms, like, for example, self-healing or self-attaching and growing? And this is an idea we had been very interested in exploring for that first project with PS1, but we decided for a number of reasons to stick with something a little more predictable. But then we found the opportunity to re-experiment with this possibility for a relatively recent installation at the Pompidou Center. And so we started experimenting with growing units of mycelium material and placing them next to each other when they were still growing and alive. And we quickly saw that we were able to achieve what we are now calling biowelding. So the living units essentially fuse together, and we have basically brick and mortar in the same physical object. This kind of time lapse can show you a sense of how it works. So you have these objects, calibrated at the right time of growing to be placed next to each other, and they can kind of fuse together. In this close-up photo, you can see kind of how it works, how the blocks attach to themselves when timed in just the right way. Here was a test of bucket-sized objects growing together and creating this kind of unusual aggregation. So this gave us many ideas for architectural experiments. And one of the first ones we looked at was combining biowelding with the process that we call voxel 3D printing. So the idea is specifically placing or 3D printing different kinds of units, in this case squares, but it's important to note here that we had two different kinds of units, mycelium units and wood units. And we had three different kinds of interactions. The mycelium unit binds to mycelium unit, as I just described. The mycelium unit also binds to wood units, but the wood unit does not bind to wood unit. So we basically have these different kinds of adjacencies, which allows us to kind of fabricate or create a building where some of the units are scaffolding, wood to wood connections, and some of the units are growing together, and we can create, a model scale prototype out of this process and using growth over time. So here you see a model scale prototype where you have this kind of shell, exterior shell of mycelium units and interior shell of wood units, all kind of self-aggregating, self-held together through this new process of both specific aggregation through voxel 3D printing and specific attachment through biowelding. And in the next line of research, we experimented with combining biowelding with something else, in this case with what's sometimes called material jamming. So jamming is this material effect that occurs when units are poured on top of one another. And you can think of units as being a variety of things, including grains of sand, as shown in this photo. So, consider grains of sand, and you're just pouring, sand into a pile. If the grains of sand are spherical, then you're going to create a pretty shallow mound. But if the grains are more angular and faceted, then you can create a slightly steeper mound. So essentially, the geometry of the grains can create different shapes of aggregating the grains. If the grains have something like a forked shape, then you could create an even steeper mound. So this is a material effect that's used in a variety of different real world cases, including in a whole science around grain silos. So think of grain silos, you're pouring grains of some kind of agriculture into a big silo, and you have a funnel at the bottom where the grains come out. So sometimes funnels can get clogged in grain silos, and that creates a big problem. So there's a kind of obscure subset of science in researching how to avoid these clogs, because a clog can be a bad thing if you have a grain silo. But we looked at this problem, as design often does, it's a technique familiar to architects, and tried to kind of flip it on its head. And we said, well, what if we wanted to create a clog? What if a clog in some kind of funnel, and we broaden out from grain silo to think of other kinds of funnels or shapes. What if a clog could be a good thing? What if a clog could help create a kind of architectural form? And that's what you see us getting excited about and inspired by on the right. What if we could purposely create a clog through material jamming? And also biowelding. So here we're exploring some new unit types and ways of aggregating some units. And then we also did a series of physical tests about clogging. So here we're exploring a certain geometry of a unit, this kind of fork-shaped unit, a certain geometry of a funnel or a kind of formwork, and a certain geometry or placement of pouring units into the formwork. And you can see that based on a few different tests, we get different results based on these combination of these factors. The form, the shape of the formwork, the location of the funnel, and these two slopes that are varying, and also where you're pouring the units. We arrived at something we thought was pretty interesting, maybe to create a larger scale architecture. And then we scaled up and changed materials. So here we're using actual mycelium units. We're using basically a quarter scale model. So we were interested in creating a 4-meter tall installation. And here we're testing a 1-meter tall prototype. And you can see that through a combination of these geometry and the material jamming effect, we were able to get an interesting type of arch created in a new kind of way. At this point, we brought our insights from our scale prototypes, our ideas about jamming and biowelding, to Paris, where we were working on site to create an installation for an exhibition at the Pompidou Center about biodesign, essentially. So how did this work? We grew a bunch of new mycelium blocks in bucket shapes. This was partly because this was a reusable formwork and easy to create using something off the shelf. We grew these units in phase 1 of growing. We took the units out of their buckets at just the right time and poured them in this full scale, basically 4-meter tall formwork. So here are the units. We took them out of their buckets, and we basically poured them in a couple of different phases into this 4-meter tall formwork, trying to scale up from what you saw in the quarter scale test. And so here are all the units poured in. Hopefully they've created the jamming effect that we wanted. We remove essentially like the plug at the bottom of this funnel, if you can think of our formwork as a funnel. And here there's supposed to be a series of really loosely packed units on the bottom, which we can just kind of shake out or push out easily. And then there are a series of units on the top that have created a more self-organized shape of a compacted, jammed structure, it's hopefully in an arch shape. Then we had our final phase, which was growing the objects together through biowelding. So we're hoping that these bucket-sized units are going to stick together through biowelding. And we remove the formwork. Hopefully they don't just pop off, which they don't here, which was exciting. And in the end, we have this, kind of human-sized arch, 4 meters tall, created out of this new process of biowelding and material jamming. And we think this suggests new possibilities for a way of creating some architecture in the future. Okay, so finally, I want to actually I'm gonna show this really quickly. I want to talk about how our this kind of applied research engages with the more traditional process of designing and constructing buildings. So here's a project that we created recently, which is called the Embodied Computation Lab at Princeton University. In many ways, it shares some similar ideas to the OHM project, which we saw earlier today. So exciting, what a great building. And in this case, this is a building for a university for the next generation of architecture research, not only for biotechnology and the built environment research, but for research on a variety of next generation technologies in architecture, for experiments with things like robotics, sensors, and everywhere that computers meet the physical world and become what we could call embodied computation. The building is located near the athletic fields and the new chemistry building. Historically, this has been a site of great architectural experimentation. It's where Buckminster Fuller made his first geosphere with students at Princeton. The program for the building involves multiple uses, from teaching to fabricating to testing, but more importantly, the program calls for the flexibility to adapt over time to new research and new equipment. In other words, we had to design both for the known research and equipment of today, but also for the unknown research and equipment of 10 or 20 or even 50 years from now. And our concept here, again, a little bit like the OHM building, was to create what we are calling an open source building. So like with open source software, this was a building that was designed to be kind of, we could say, released as a fully functional version 1, but also to be changed over time by multiple authors. And in this case, it works in several ways. First, the building was deliberately incomplete, with about 2/3 enclosed volume shown here on the left and 1/3 open frame on the right. Second, the building has many swappable modules and systems, such as radiant heating that on day 1 draws from the waste condensate of the building next door, essentially heating the building for free with no additional energy consumption or carbon emissions. But in the near future, in a kind of plug and play system, the building can start drawing heat from a geothermal well that the faculty researchers are planning. Third, the building is rewritable over time. So just as a biologist uses a microscope to study organisms, we hope that architects will use this building to study buildings, continually adding to it and adapting it. And fourth, this is a building that learns over time. So like living organisms, it continually processes information, including high resolution data from sensors for things like heat and light and occupation, and it evolves based on changing conditions. The building also has several unique sustainability features, such as a floor for both radiant heating and radiant cooling. Here you can see it in winter with heat as one of the invisible but important features that's being monitored continually in the building. It has a structure made of engineered timber or glulam beams and columns rather than steel, even though there are some pretty demanding structural requirements like a 5-ton gantry crane. And the facade is made of repurposed scaffolding boards from New York City construction that would otherwise end up in a landfill. So scaffolding boards are these 2-inch by 10-inch planks that are used in every construction site in the city. They're typically used for about 1 year and then literally thrown in a landfill because some of the boards have warping or cracking, and it's easier to just create a rule of thumb, like throw them away, than to look closer. And with our use of material, this kind of salvaged material, in a way a biomaterial of aged wood, the final layer of constructing the building also became the first experiment in operating the building. So in other words, we started with these weathered scaffolding boards as an interesting biomaterial that's non-uniform. We developed the hypothesis that wood with micro contours might actually offer things like thermal benefits through trapping small pockets of air and acting as an invisible jacket or insulator. And it also might offer hydro benefits through shedding water. So we knew that the material might offer these performance benefits. And we also knew that different zones of the facade of this building, like any facade, might have different performance requirements, as this solar analysis shows. And we knew that we had thousands of different boards, with each one with different properties to arrange on this building facade. So which board with which properties should we put in which position on the facade? And at this point, we turned to a version of artificial intelligence to help us manage this complexity and fine-tune the design. So we knew that areas of the wood with the narrowest grain provided the best performance, and we knew that the regions around knots in wood tended to correlate with this narrow grain that we wanted. So we set out to identify and treat the knots in each scaffolding board through these two different techniques. One is machine learning, and the other is CNC sandblasting. Machine learning is a technology that is important in our world. It's influencing many things for both better and worse, but it's a technology that architecture has been a little bit slow to experiment with. And so we wanted to try out what would machine learning for the physical world look like. Sandblasting is a fabrication process that can create a type of artificial aging or weathering, and it's relatively underexplored in most digitally driven architecture. So unlike something like CNC milling and most other forms of fabrication, sandblasting enhances the variation in texture and materials rather than just smoothing them over. So with sandblasting something like wood, sandblasting erodes the soft areas and preserves the hard areas, resulting in complex pattern and in the kinds of micro contours that we were interested in for building performance. So to apply machine learning and CNC sandblasting to the facade of this building, first we took all of our boards, several thousand boards, and we photographed all of them, creating many thousands of square images of wood, like the ones shown on the screen here. And then we used a little bit of human training, asking people to look at these photos and click either not with the KN or not with an N. Look at the photo and determine whether it has a knot. And after some initial human training, the machine algorithm got pretty good at kind of seeing where the knots were, even in photos that had never been seen or classified by a human. And here you can kind of see it at work, what the machine thinks of these knots. Here's a different view of the algorithm at work, from left to right, kind of honing in time on where it thinks the knots are. And it was fascinating and even a little bit scary that the human had simply stated whether each photo had a knot, but the human didn't circle the knot or describe it in any way. And the computer was able to look at a new photo of wood and not only figure out whether the photo had a knot, but also find the location of the knot, of that unique feature. So after using machine learning to identify the knots, we invented this custom robotic sandblasting machine to create micro contours exactly where the algorithm told us there were knots. The process for each board looked like this. We created some custom machine and control software that looked like this to do the CNC sandblasting. And overall, this allowed us to explore a new approach to materials that we could say is both computational and biological. And we applied this new process to arranging thousands of boards such that the performance was best suited to zones on the facade based on solar analysis. For example, we were trying to provide more thermal insulation in zones that were most shaded and that needed it most. So this was, of course, a complex computational problem. It was interesting as kind of computational research to find the design that best matched our goals. But probably more profoundly, this process allowed us to celebrate variation and non-uniformity in materials rather than erasing it. An approach that we think is more generalizable to all of biotechnology in the built environment, for example. So it allowed us to see how each board comes from a specific tree that grew in specific conditions and formed in specific ways, and to think about how we might use pieces of materials in relevant, suitable ways, ultimately overturning our kind of default rules and lowest common denominator approach to building materials. And in this respect, the facade kind of registers the entire story of the material, from resource extraction from a tree to initial use as scaffolding boards, to computational analysis of unique features, to selective fabrication through sandblasting, and eventually to reuse as a building facade. And more fundamentally, this allowed us to develop a kind of a new perspective on all buildings as just a temporary formulation of materials, energy, and labor connected to other formulations before and after the life of the building. So the design registers at these different scales. Here you can see a close-up of the board and the 3D topography. Here are the boards installed on the building. Here is the facade itself as an example of what we could call embodied computation of the current state of artificial intelligence and machine learning 2020 with all of its errors and mistakes and promises and risks. Here's a view of the gantry crane in action. Here you can see the two large hangar doors on each side of the building that open up and adapt to different research scenarios. Here's some of the research equipment in the building, including these multiple industrial robot arms on tracks that collaborate with one another. And in the end, this dynamic and adaptive structure, we think, proposes an alternative to buildings that are static and driven by form or aesthetic signature alone. The building suggests a new hybrid design approach that's high-tech and low-tech, a familiar and new functional and aesthetic, digital and biological. And while the building itself is an open-ended research project, it's also a tangible demonstration of design, of atmosphere, of the experience of space, of what it might feel like to live in a strange new architecture designed for both humans and machines in the context of the natural environment, with this vision for a new form of living architecture. Okay, so finally, to kind of come full circle, and I will keep this brief, but I'd like to show this line of research that's kind of still in progress, and it goes back to some of the applied research logic of the previous projects. In other words, this relationship between research and actual buildings is not linear, it's a cycle, it's like feedback in both directions. And so this is a project that begins with the physical material from a previous project with sandblasted wood, which you can see here, and with an open question about what possibilities the material might open up, what kind of performance we might get out of it. And in a different way, this project begins with new ideas about ecosystems and public health, about some of the same things that we've been talking about in the summit over these amazing 4 days. So this is a project with two good friends and colleagues, Kevin Slavin and Elizabeth Penoff, and it builds off some of the amazing work that they and others have done, and some of you and the past presenters have done at the intersection of biology and cities. This is for an installation at the Storefront for Art and Architecture in New York City. And here we set out to explore what we could call two concepts, maps and models. For maps, we wanted to measure the microbiome at a gallery in Soho in New York City and compare it to the microbiome of places like the Brooklyn Navy Yard in a different part of New York City. And for models, we wanted to explore design scenarios for architecture that promotes microbial life. And we were thinking about whether something like probiotic architecture might actually be able to improve public health, the kind of things that you are already doing and working on and familiar with. In our case, we worked on a version of a bioreceptive material to catch and host microbes deliberately. And we started with wood, as I already mentioned. Wood is well suited for hosting microbial life based on its molecular composition and its micro and macro shapes. And at this gallery in New York City, we transformed the panels of the exterior facade with wood tiles cut from standard lumber. Then they were deliberately eroded through sandblasting at various depths to create diverse microclimates. Each microclimate had distinct grain and knots that created distinct pockets of shade and moisture. And we and this kind of approach to materials as a continuation of the previous project allowed us to see that even the most common and humble building material, a cheap 4x4 post of Douglas fir, has a material story that could be uncovered that could offer a range of visible and invisible effects. Like all our projects, we did a series of material tests, assembled ourselves in new ways. And then in terms of these physical materials, which you can see here on the building facade, with the building envelope of this bioreceptive wood, we think that architecture could become this kind of sensor for the urban microbiome. For the second part of the project, in terms of actual living materials, we set up a lab inside the gallery to sequence DNA on site. And here were the basic steps. We swabbed sandblasted wood tiles from each location, several different locations in the city, to collect the surface microbes. We placed the swabs in a liquid solution to break down the cell walls. We separated the DNA from the other substances. We added marker beads to the ends of the DNA fragments to produce a pure DNA solution. We loaded the DNA solution onto a flow cell to be processed by this portable DNA sequencer, amazing technology that allows you to do this like just on a laptop. And the sequencer kind of converted the electrical charge into the sequence data, the ATCG of genetic code, which you can kind of see here. So finally, after sequencing the DNA from several different locations, we designed a system to automatically draw out the output as an early and rough map to this uncharted territory of the urban microbiome. So of course, this map is raw and at a coarse resolution, but it's already allowed us to see some interesting things. For example, in the Brooklyn Navy Yard, we saw microbial DNA that performs the function of breaking down heavy metals, which basically indicates that the Navy Yard has specific pollutants that were not found in Soho. More generally, we're using a gallery as an active living experiment. We're exploring this new kind of architectural envelope and a new way of seeing some of the important matter and organisms that help keep us alive. We're looking at materials and their layer of living material, reframing buildings as stewards for the urban microbiome. The initial evidence indicates that we are all connected in our kind of collective future, all humans and non-human animals and microbes living together. Which will give me the chance to show one last thing. I know I'm running a tiny bit long here, which is a current project that we have at the Venice Architecture Biennale that is up now. So this was directly a continuation of this line of research of the urban microbiome and bioreceptive materials. But we wanted to take it to a different direction, exploring a new bioreceptive material and a new pavilion as a prototype of multi-species buildings and of probiotic architecture. So I'd like to conclude by offering a kind of story about the future of living buildings that we hope is both exciting to architects and also relevant and interesting to the general public. Microbes are everywhere. Invisible to the human eye, they live in the air, within the walls, and on even the cleanest of surfaces. We have always lived together with microbes, and the vast majority of microbes contribute to our survival and our well-being. The human body has more microbial cells than human cells. Although a few types of bacteria and viruses can make us sick, many other types actually defend us and limit the effects of the harmful ones. Bacteria in the human gut help us digest food. Viral DNA in humans helps us process and store memories. And from this perspective, each of us is a metropolis teeming with life. And like the residents of the city that contribute to its culture and make it run, the microbes of the human body contribute to its vitality and give it energy and personality. Microbes help make us who we are. As I read this description, you will shed 2.5 million bacteria, breathing them out and shedding them from your skin and hair. Most of them will die. And the ones that survive in our typical architectural environments, the arid landscapes that we heard about at the beginning of the summit, tend to be the ones that have antibiotic resistance and enhanced virulence, the ones that are most harmful to humans. Yet in environments with a high diversity of microbial life, the harmful organisms tend to be outweighed and mitigated by the benign ones. So what if we could change our architectural environments to be better hosts for a diversity of microbes and to decrease the amount of harmful ones? In this vision of the future, architecture could include spaces for both humans and microbes, Buildings could be designed as microbial environments. Rooms could have different microclimates and many types of microbes. Equipment for ventilation, heating, and cooling could be supplemented by microbial reservoirs. Materials could be sponges, textured, porous, and organic with infinite surface area. A bioreceptive material like loofah could be designed to catch and host microbes with tiny cavities of different sizes and shapes to create different pockets of shade, temperature, moisture, airflow, and nutrients. And with walls of bioreceptive material, architecture becomes a cloud to nourish humans, and in turn, buildings become stewards of the microbiome. Just as a healthy gut microbiome promotes our individual health, a healthy urban microbiome might promote our collective health. And a new kind of living architecture might involve these probiotic buildings. A holobiont is a collection of diverse species living symbiotically, each contributing to the well-being of the whole. The humans, microbes, and other organisms in the holobiont are mutually supportive. In this way, we are all made of each other. This gives us a new way of thinking about multi-species architecture. In the future, buildings with porous surfaces could behave like our skin and gut and support colonies of microbes tuned to create healthy spaces organically and inexpensively. These buildings might look very different than the ones we currently inhabit, opening doors to new ways of thinking about how we live and how our cities are organized. Humans are the microbes of the city. And with microbes, as with humans, diversity is the key to well-being. To design for diversity is to design for living together and to design for our collective future. Thank you.
Thank you very much. That was absolutely fantastic presentation. It's given us a lot to think about. There are a lot of questions in the chat. I think we'll take some of them, maybe carry on until 10 past, and then just have a very quick break before the next session at 15:15. So, I mean, there's some quick fire questions to start with. In fact, the first two questions from the audience were exactly questions I had in my mind, going back to hi-fi. Just some quick questions about, did you have to join the mycelium bricks? Did you bond them in some way?
Yes, and great question. With hi-fi, we joined the bricks not through biowelding, which I described for the later project, but through a relatively sustainable mortar. So we used the same type of bricklaying technique used in traditional architecture, brick architecture, with brick masons and with mortar and with strings and leveling. But in this case, we used a mortar with almost no Portland cement and a lot of lime in order to kind of hit the right calibration. And we actually, without going into all the details, we did a lot of testing along these lines. In other words, we needed a mortar which was strong enough. Traditional mortar would have been perfectly fine, strong enough, but we needed a mortar that was compostable. So we made a series of different mixes and tested them both for strength on the one hand, one kind of performance, and for compostability on the other hand, working with different collaborators. The strength of materials people just wanted something strong. Our structural engineers wanted something strong, but our composting collaborator wanted something that was compostable, and we were able to find something in the middle.
Yep, nice. And of course, that's where the biowelding is perfect, because it's always that bonding that causes the trouble. And if you can just eliminate that, then you get a much more elegant structure.
Exactly.
And then another question about hi-fi, about how long do you think the structure would have lasted outside before it would have started degrading in place, and whether the blocks had any sort of surface coatings to protect them from the weather?
Yeah, this is another really important line of question. So when we were doing some of our initial testing, we took some of our bricks and subjected them to what's called accelerated aging testing. And this is like a chamber that you can use to artificially accelerate the weathering process. So some of our bricks went through what we could consider 3 years worth of New York City summer. So 3 years straight in a row of summer, with the conditions of UV exposure, rain and drying, variation of humidity, 3 years worth of what's typical in New York City for the summer, and they still have the exact same mechanical performance, the structural performance. So that's evidence that, this is a pretty robust material. It's compostable when you break it up and when you put it in a different scenario, but in its built scenario, it's pretty robust. We only left it up for 3 months and then took it down. It basically behaved as we expected, but we do know that the weakness of the material, exactly as the second part of the question suggests, is in the coating. So if, for example, you had taken a pencil and stabbed one of the bricks, that would create a problem. So in terms of robustness integrating into today's buildings, I think there both would need to be some more calibration and engineering of the material, but also potentially what all buildings of today do, other layers of materials. So we actually had a project that we were working on with some amazing collaborators for COP25 in Chile, which eventually got canceled and moved, where we were creating a series of pavilions outdoor with mycelium bricks that were coated in plaster, or to test a variety of coatings. And we strongly believe that if we had coated these bricks in a material like plaster, potentially a sustainable or compostable plaster, that they could last, for dozens of years, just as they are with our existing bricks. But at the same time, I also like to mention that, probably different people in the summit have heard this kind of thing or thought about it or are working on it. That if we were just to have more time to work on mycelium materials and calibrate them, we could make great strides in making them more robust, more weather resistant. And one thing I sometimes think about is that 3M, the big materials company in the United States, but globally, I think, has at one point invested in mycelium materials as what they called a materials platform, like plastics are a materials platform. In other words, you can create all kinds of plastics, thin, flexible ones, dense, rigid ones that are like fabric, ones that are, like a foam. And mycelium potentially has the potential to do that too. As we know, we're seeing mycelium leather and things like that. So I think, we can combine mycelium with other materials, definitely today increase the life 50 years and beyond. But we could also continue engineering mycelium materials to be more robust and have a variety of other performances. And I think, it's so great that programs like yours at HPBE are working on this kind of thing, doing these experiments, testing them out in the real world on that amazing home building.
Great, thanks. So a question here, which relates closely to some work we're doing actually, is could you elaborate on how the Pompidou Center reacted towards having living mycelium in its gallery? Because we're having issues of this as well, of proposing to make large mycelium constructions within the home, but then issues of, is that going to start eating the structure of the building? Is it going to get onto the wood? What are the health issues? So I'd be very interested in them.
It was definitely a discussion and an unknown or something that was beyond what is normally in those galleries in the Pompidou Center. In the end, they were very excited about the show, about doing something full scale, about an exhibit that would not just show things that had already been tested somewhere else, but that were tested for the first time in their gallery. Their biggest concern came from the line of thinking of their kind of conservators about if there were spores that were being shed from the structure, especially in the case that we were deliberately not drying out the material until a certain time. So if there were spores, they were getting nervous. And so we had a lot of discussions about how to ensure that there were not spores, how to ensure that we were not going to somehow shed some microbes that would then start eating other artwork or causing problems. We did see, though, by the way, that with our hi-fi installation, which was a different scenario because it was outdoors, but there had been some concern about allergies potentially. And we saw that there was not a single, instance or example of people kind of reacting negatively to this material. And although it was outdoors, when you were inside the mycelium structure in PS1, you felt the material, you could smell it. So it was there. But in essence, I think like a lot of new ideas in architecture, it's going to take some pioneering work by the, museum or university or architectural client who's willing to test it out for the first time. And of course, be careful, not create health risks, but to be, willing to do it for the first time.
Yeah, thanks. It's interesting. A couple more quick questions. I know I said I'd stop now, but it's too interesting to stop. A very nice short question from the chat. How do you get funding for this stuff?
Any way we can, essentially. I mean, so some things like the PS1 was a competition, and it's incredible that the Museum of Modern Art has run that competition for many years and gives a platform and a budget to a young firm to do something. The Pompidou was also a commission, and Venice was I guess a commission as well. But in some cases we definitely are trying to convince more typical clients, like a university, as you guys are doing, to take a risk and do something more experimental and unusual, which we have done with Princeton, we've done with some other places, including we're working with Airbus on the design of a factory building, where I think increasingly people understand that the do nothing approach, the status quo approach, has risks as well. So if we just keep building things as normal, it's not like that comes with zero risks. There are different risks. Some of them are longer term, some of them are not on the exact site of the building. So I think increasingly, as people are aware of climate change, are aware of the need to reinvent our building materials and our built environment, there are going to be more people who, including clients and funders, who are willing to say, hey, we need to do something. And this idea of healthy buildings, there are a lot of ways to do that, and we should be funding them and exploring them and commissioning them for a building.
Yeah, thanks. And then finally, my question, I was wondering if you see a conflict between computational design and craftsmanship. I was thinking for the hi-fi and the sort of computational layout of the bricks, which perhaps could have been sort of handed over to expert bricklayers if they'd had a selection of sizes and could have worked it on site, but also the timber facade with the sandblasting of the boards. And the construction becomes must have been quite a laborious, painful process, doing it by number rather than sort of selecting boards on site and allowing the craftsman to have that creative input. So I was wondering where you see the balance between those.
Yeah, I think that's a really interesting line of thinking. And I guess I definitely see it, I don't quite think of it as a conflict, but maybe a productive tension. I mean, I think ideally we can start thinking about leveraging the best of both, or a kind of multiplier effect, or a new possibility that emerges when combining these things. So, the digital and the biological, or the craftsman and the computational, could be seen as opposite ends of the spectrum, but they could also be hybridized. And I think we're most interested in looking for these new hybrids that can leverage the best of human creativity or the best of craftsmanship knowledge, with the best or a new twist through computation. So we are often, just more tactically, we're often aware of not, even though we're very interested in computation, not letting computation drive the boat, so to speak. We don't want to lead with that. And that's where, like, as I was kind of showing with hi-fi, we turned to computation when we had no other way to figure out how to lay out these 10,000 bricks and make the shape and make it predictable. So we were using it to solve a problem that we knew. We sometimes use it in a more generative way, but we always like to be in control of it, not letting it control us.
Yep.
And I think we could say, interestingly, the same thing about craftsmanship and about biology. So not just, so we did have to, we went, so to speak, to the computational architects and said, this is not a building about computational architecture. That's not what we're trying to experiment with here or show here. We went to the brick masons and said, this is not a building where every course is directly on top of the other. It has these offsets. It has this unusual form. And I think similar with biology, we're saying like, hey, we can do a lot of things with biotechnology, but what do we want to do? What's driving that? What are our reasons for that? Even if sometimes they're open-ended experiments, but making sure we fit the whole package together. And that's kind of why I begin with that diagram of all these interconnected loops. We've got to design the whole thing together, the package of computation and biotechnology and craftsmanship and society. Like, what are the ways that we can put all those pieces together in different projects?
Fantastic. Thank you very much. We're over time now, but that was a really nice way to finish, looking at those interactions and talking about how everything needs to work together. So